Hello, my name is Perla Tovar. I am a senior in biochemistry major here at SFA in the Department of Chemistry and Biochemistry, and I have been working with Dr. Sengupta on a project titled The Influence of Phytochemicals Present in Mint Extract in Helping Prevent or Reduce Oxidative Stress in Human Serum Albumin Protein, or HSA. Now, the overarching goal of our study is to perform multiple drug screening on multiple mint extracts in order to see which one might be more potent in helping reduce oxidative stress and possibly develop a drug against a disease. Now, in order to do this, we kind of had to simulate what happens in the body. And specifically in the body, oxidative stress can build up. And this buildup is caused due to the formation or accumulation of free radicals, which are unstable molecules in the body. And when these free radicals accumulate and there's more of these than antioxidants, which are stable molecules, it can cause damage to target molecules such as membrane lipids or lipid membranes, our DNA, or protein. Now, in order to stabilize these free radicals, we can use antioxidants to help reduce oxidative stress and further help decrease the probability of developing certain diseases such as cancer, Alzheimer's disease, Parkinson's disease, or diabetes. Now, we know that from literature that mint plants contain phytochemicals, which are compounds produced by plants, and these phytochemicals can have antioxidant and anti-inflammatory properties that can help stabilize these free radicals and help reduce this oxidative stress. Now, for this study specifically, we have been using 3 mint plants which were grown in my advisor's garden, and they are peppermint, chocolate mint, and spearmint. Now, to continue our study, we had to see whether or not our oxidative stress or our free radical was being formed. So in order to do this, we used the methylene blue dye test, which basically, if bleaching occurs, that means that a radical has been formed. And so when we conducted this study, we saw that when we reacted both hydrogen peroxide and ferrous sulfate, which are Fenton reagents, that the methylene blue dye strip, it bleached, indication of the formation of a radical. So after we tested this out, we conducted multiple other studies using different techniques in order to see the effects of oxidative stress on our protein, and in the presence of our mint extracts and without the mint extracts. And so one of the techniques we used was SDS phase gel electrophoresis, which by quantitating or quantifying the band intensities, we were able to see the effects. And so when our protein was induced with oxidative stress without any mint extract, we saw the most change by seeing that the band intensity decreased the most, indicating that our mint extracts possibly were helping reduce this oxidative stress or damage. And specifically, when we induced our HSA in the presence of our mint extract, specifically chocolate mint, this band intensity decreased the least, indicating that our chocolate mint was a more potent antioxidant than both our spearmint and our peppermint. Now, we will further our studies later on to keep looking at the effects using different techniques, and we will also further our studies by studying these effects on mammalian cells. And thank you for listening.